video explains how to do RNA extraction on swab samples collected for COVID-19 diagnostics. You will need RNA extraction kits, swab samples collected into lysis buffer, 1000 microliter and 200 microliter pipettes, pipette tips, a discard, and a microcentrifuge. A biosafety cabinet is recommended but not essential. So let's get started. Before bringing the sample bags into the biosafety cabinet, spray them with 70% ethanol. Once inside the cabinet, spray the inside of the bags and the tubes with ethanol. This should kill SARS-CoV-2 virus on these surfaces. Take out the RNA extraction kit buffers and place them in the rack. Start by pipetting 500 microliters of 100% ethanol into each tube. Incubate the tubes at room temperature for 10 minutes to kill any viable SARS-CoV-2 virus in the sample. Get out all the tubes you need for the rest of the extraction. You will elute the RNA into 1.5 ml Eppendorf tubes, so make sure to label these tubes very clearly using alcohol-resistant markers. Now take the lysis buffer out of the fridge. First, add 400 microliters of lysis buffer to each tube. Then transfer 600 microliters of each sample into an RNA spin column. While doing this, make sure you're changing the tips between tubes. Now open up the centrifuge and remove the entire rotor by undoing the bolt with the key. Make sure you open the rotor in the cabinet. Put your samples in the rotor and close the lid. Spray down the rotor with ethanol. Take the entire rotor over to the centrifuge. Tighten the bolts. And close the lid. Now centrifuge for 30 seconds at max speed. Make sure to take the rotor back into the hood before you open it. Decant the liquid into a waste container. Make sure you dispose all liquids as chemical waste. And place the column back into the collection tube. Now transfer the remaining amount of sample to the spin column. Place the spin columns back into the centrifuge rotor and centrifuge for 30 seconds at maximum speed. Decant the liquid and place the column back into the tube. Change the pipette to 500 microliters and then add 500 microliters of wash buffer to each column. Place the tubes into the centrifuge rotor and spin for 30 seconds. Decant the liquid and place the column back into the tube. Now we add 500 microliters of wash buffer 2 to each sample. Place in rotor and centrifuge for 30 seconds. And decant the liquid. Now repeat the wash buffer 2 step by adding 500 microliters of wash buffer 2 to each sample. Centrifuge again, but for 2 minutes. Decant wash buffer 2 and discard the collection tube. Now place the column into a new collection tube. Add the collection tubes into the rotor. Centrifuge for 1 minute to ensure all ethanol is removed. Decant and discard the collection tubes. Then transfer the columns to clean 1.5 ml sterile Eppendorf tubes. Now elute the RNA from the column by adding 100 microliters of RNase-free water. After you've done them all, leave the columns to stand for 1 minute. Then centrifuge for 1 minute at maximum speed. Now take the tubes out of the centrifuge rotor and discard the columns. You should now have 100 microliters of extracted RNA ready for qPCR or sequencing. These samples should be stored at 4 degrees if you're going to use them immediately, or at -80 degrees for long-term storage.